Hey, hi everyone. Happy Friday. Um, let's continue about talking about our innate immune system and sort of continue from where we left off. Remember, we said that our innate immune system consists of sort of all of the pre-programmed knowledge of how to produce certain cells that can recognize and protect against pathogens or even diseased cells inside of our body, right? Those effector cells can recognize those to protect us against disease. And our innate side also represents all the genes that have the know-how to encode for things like complement proteins that can then attach to the surfaces of pathogens, opsonizing them, making it easier for things like our innate immune effector cells, like macrophages, to go ahead and stick to them to eat them more efficiently. And as we also learned, that those complement proteins can proceed to poke holes in the surfaces of bacteria or even viruses, causing them to directly be attacked and destroyed. Right? So those cells and protein components that make up our innate system, right? We call them innate because you are born with that know-how, right? All of that knowledge is encoded for by the genes that you inherited from mom and dad. So even within that innate response, we said there are two sort of stages to our innate immune system. The immediate response that involves things like those macrophages or complement proteins that happen to be at the site of the tissue when you get something like a cut, and right away, immediately, right, have a chance to start attacking the pathogen and detecting it. And there is what we call the induced response. That is to say that if that combination of macrophages and soluble proteins like complement proteins can't take care of the infection within a relatively short period of time, maybe a few hours, right? Then maybe those macrophages need to call for some backup to help them out, right? And that is what we call the induced response. Keep in mind, right, the induced response will still involve effector cells or different proteins that are still part of the know-how that's encoded for by your genes, that you've had since the day you were born. And so therefore, we're still talking about innate immunity. All right. So let's think about it this way. The immediate versus the induced innate responses. So the immediate happens when, okay, you get something like a cut, right? So for example, a pinprick, right? Small little pinprick. But hey, if you were to look at the tip of that pin, so this is an electron micrograph looking at the tip of the pin, you'd actually see that it's all covered in bacteria, right? So you might have thousands of individual little bacteria that are on this pin, right? And so the moment they enter your tissues, right, that is the moment that infection starts. Remember, as immunologists in training, right, we have to refine our definition of when infection actually starts. And very simply, it's just the moment that you have pathogens breaching your sort of physical barriers, like your outer skin, for example, or your internal mucosal layers, and actually getting access to the underlying tissue. That's the moment when infection starts. So right away, what do we see here? So the immediate innate immune response would maybe represent the first few hours following the moment that infection takes place. So during this time, you have a pinprick that happens. So pathogens invade the tissues and they begin to proliferate, right? These bacteria, boy, they're super happy, right? They were hanging out on this pin, like on the ground or something like that, right? And that didn't really provide them a good environment to carry out their life functions. But all of a sudden now, great, they're inside your tissue, right? Where there's plenty of water, the pH is great, they're protected, and importantly, there's lots of nutrients in the form of your own tissue and cell components, right? So the bacteria start to multiply. You'll have macrophages, so certain phagocytic cells like macrophages that are already resident in your tissue, meaning they happen to be there. They go to work, right? As well, the pathogen is recognized by preformed soluble effector molecules, right? Things like complement. Right? So we have complement coursing through our blood, and a lot of complement is already kind of leaching out of your blood into your tissues. And so whatever complement proteins are there can start going to work. Right? Opsonizing. So here we have C3b. Remember, complement protein C3 is probably the most important one. It gets activated, and the C3b part of it starts to coat the surface of the bacteria. And this is like the Velcro, right? The Velcro that then makes it much easier for the beach ball or the macrophage here to be able to easily hang on to the ping pong balls, right? And eat them more efficiently. As well, we learned that the activation of C3 and the fixation of C3b on the surface of the bacteria can actually give rise to the activation of C5, right? And then C5, C5b, more technically, recruiting C6, 7, 8, and 9 can form these membrane attack complexes, right? So at the same time that you have millions of C3b spreading across the surface, right, you also have the appearance of lots of holes, these membrane attack complexes that directly then destroy the pathogen, right? So that combination of the macrophage eating stuff and the complement proteins helping the macrophage eat stuff and poking holes in the bacteria, right? A lot of times, most of the time, that'll take care of the infection caused by this pinprick, right? So the pathogen is eliminated, the infection ends, and there's very minor tissue damage because the pathogen never really had a chance to cause a lot of damage, right? So that's why if you step on a pin, right, and you're like, and you pull it out and maybe there's a little blood or whatever, right? I mean, chances are by the end of the day, you've forgotten about it, right? Because this immediate response probably took care of it. But what if for some reason the immediate response isn't enough and the pathogen, like this bacteria, somehow outruns the immediate response? Okay. So the pathogen can outrun the immediate response if, for example, too many microbes got into this wound, right? So maybe you're not talking about just this little pinprick or a paper cut, right? Maybe you're talking about something a little bit more serious. So you have a larger wound, right? Or let's say that this wound is actually a breach of the lining of your GI tract, for example, right? So for example, if you end up having, let's say, some pathogenic bacteria, bad E. coli that you got that was hitching a ride on some undercooked fish or poultry that you ate, for example, like salmonella, right? It can start damaging the cells of your small intestine lining. And as a result, you might then compromise that physical barrier. And suddenly you have pathogens potentially flowing into your tissues in great numbers, right? It could also be that the microbes themselves might be poorly recognized by things like complement proteins and macrophages, right? Maybe something about the chemistry or the structure of the cell wall of the pathogen makes it harder for complement proteins to attach, and therefore harder for things like macrophages to effectively eat them, right? It could also be that the microbes themselves, once they start getting happy in your tissues, right, are secreting different types of proteins or other types of molecules that actually deter or interfere with immune cells being able to effectively recognize them. We'll look at examples of those later on. But the bottom line is the immediate innate response can't keep up. Right? So the pathogen is not eliminated, and therefore you have to call for some backup help. Right? So that backup help then is your induced innate response. So the induced innate response would sort of be like if you still have an infection after a few hours up until maybe a few days, right? This is when you'd really start seeing the induced response reflected in changes in the physiology of your tissue at that area. Right? So Um, what we're applying here is that somehow the immediate response didn't work, right? So if you follow this flow chart, you're sort of, and now at this point, right? So the cells in the infected tissue are being activated, right? Many cells are being attacked and damaged. Therefore, there's all kinds of messages being sent out by not only the macrophages themselves that are busy still trying to eat stuff and sending out messages, right? But the actual cells of your tissue that start getting damaged, they send out messages as well. What kind of messages? Remember, we're talking about small proteins that get activated inside of these cells, right? Proteins that are coded for by genes in your genome that normally might be shut down because you don't need to have these cells making these signals. But when you have cellular damage happening or the macrophages are eating things, right, then these genes get turned on and these small protein products get made. And these protein products, we've already learned, right, are called cytokines. Right? The cartoons normally in the book show the cytokines as these little triangles, right? But they're little proteins. And so we'll take a look at that today. What do these cytokines do? We already said that they do things like recruit more macrophages to the area, and they start the inflammation response. Right? So inflammation, hey, you know inflammation, right? So look at this cut here, right? You wiped out on your skateboard and you got a nasty kind of scrape, right? So a day later, what do you see? It gets kind of puffy, right? It feels warm to the touch. It might actually feel firm to the touch. Right? That's all part of inflammation. And so this inflammation is actually caused by your immune system now shifting over to this induced innate response. So what does that do? Right? So it's going to bring in more effector molecules like complement proteins and more macrophages. It's also going to bring in other types of immune cells like neutrophils to help out, right? Hopefully then, after that happens, with that added backup help, the pathogen is eliminated and the infection ends, right? But of course, the consequence of this is you're going to have more tissue damage than just simply, a pinprick or a paper cut, right? You might actually be left with a scar or something like that. Right. So familiar symptoms of an induced response, that is when your innate response has now shifted from the immediate to now recruiting some backup, the induced, is you'll get inflammation, like localized inflammation at the site of the wound. Certainly you feel pain, right? Because the wound gets very tender to the touch. Or you might have more systemic types of responses, like fatigue. You might actually get tired, right? And you might develop a fever. So, as I said, oftentimes it's easy for us to blame something like inflammation or pain or fatigue or fever on the pathogen itself, right? Oh, the bacterial infection, it's making me hurt in that area, or it's making it inflamed, or it's giving me a fever. When in reality, no, right? Because think about it this way. Microbes, the pathogen, they don't gain anything necessarily from giving you a fever or making you tired, right? It's actually your own immune system that is doing these things. These things that we associate as being bad things are actually good things, right? Because they help your immune system, right, fight the infection better. For example, the puffiness, right, and the firmness of a local inflammation represents the tissue trying to reduce the amount of fluid that is actually leaving the area and going back into your circulatory system, right? Because what you're trying to do is prevent the bacteria from actually getting into, for example, your main circulatory system. So as a result, you get some fluid buildup, and that's the firmness, right? Things like the warmth, right, is localized increase in temperature of the tissues to try to make it uncomfortable for the microbes, right? The microbes, like the bacteria, who would love it. Your normal body temperature of 37 degrees Celsius or 98.6 Fahrenheit, right? That's what the bacteria love as well, right? If you can make it warmer in the area, you can slow down the bacteria from spreading, right? Or multiplying, right? Systemically raising your whole body's temperature up right, does the same thing. Right? So if the bacteria is potentially now getting into your blood, right, your whole body raises its temperature to try to make it less effective for the bacteria to multiply. And interestingly, fatigue, getting tired. Right? These types of immune responses are more expensive for your body to deal with rather than just some complement proteins and macrophages at the site of infection. Right? So therefore, evolutionarily, we get tired so that we will be encouraged to just lay down and go to sleep so that our body's energy resources can be focused on fighting this infection. Right? Okay. So these responses, surely you can tell, are more expensive than simply the immediate responses. And that's why our immune systems don't just start with this, right? I mean, quite frankly, it would really suck if you like got a pinprick or a paper cut and then you just get a fever every time, right? No, right? That's unnecessary, right? So that's why our induced response comes after our immediate response, has failed to work in the first few hours or so. All right. So all of this stuff is mediated by cells being able to receive signals, right? And these signals might be, for example, the cytokine signals that are being released by cells to talk to each other. Or, of course, it might be small signals that are coming off of the pathogen itself. So not surprisingly, if there are many sort of generic kind of features of something like a bacteria or a virus, right, then over eons of evolution, as our immune system has evolved, our immune system has become very good, right, at looking for those generic signs. So that's what we're talking about when we said mall cop, right, is just very modestly trained to look for certain generic clown features, right, because those features over evolutionary time, right, are pretty generic types of signals. Right? So these macrophages then, like mall cop, who are doing the kind of patrolling and looking for things, right, they have to be able to recognize things by way of using different receptor proteins on the cell surface. So let's have an introduction today at some examples of different types of cell surface receptors, right, that are involved in recognizing the presence of pathogens and also, right, recognizing, for example, where certain infections are happening in the body so that immune cells that are traveling through your blood, right, can actually know where to stop and enter your tissue and help out. There's over 100 different types of receptor proteins that immune cells use. So each type of immune cell expresses just a subset of these. Right? And that subset, right, gives a population of immune cells, right, a lot of variation in terms of certain cells being good at detecting certain features better than other cells can. Right? Even within a specific cell type, like if you think about the entire population of your macrophages that are in your body, right, not every single macrophage will have the same assortment of different types of receptors on its surface. Right? Because if you have 100 or so different types of receptors, you would imagine, right, you're not going to have one cell that expresses all 100 types on its surface. Right? That'd be too much work for that cell to maintain. And the number of each specific type of receptor on its surface, if you always had to have all 100 different types there, right, the actual number of each individual type of receptor might be too few on the surface of the receptor of the cell to really be effective. Right? That's why our immune cells typically will have a handful of different types of pathogen sensing receptors, right, and just have a lot of those different types on a given cell. But as an entire population, right, then a whole bunch of macrophages can recognize different things and work together. Right? So again, if we use the mall cop example, maybe an individual mall cop is trained to recognize the rainbow hair of the clown. A different mall cop, right, is better at recognizing the white face paint. And then a third one is better at recognizing the colorful outfit of the clown. So the three of them together, right, work together to coordinate an immune response because each of them alerts the immune system that they see what they sort of have been trained to recognize. All right. So here are some examples that we'll look at today. So look at Mannose receptors that bind specific sugar groups. And there are other examples like these scavenger receptors that can bind to things like lipids. I mean, we already looked at examples of receptors like complement receptors. That's the very first receptor we talked about in this class, because when we use the beach ball and ping pong ball analogy, you stick the little microscopic Velcro hook pads or whatever to the ping pong ball, right? But the beach ball itself needs to have the fuzzy part of the Velcro so that you can actually stick, right? So we said the macrophage has to have these complement receptor proteins, right, in order to actually recognize complement C3b that's opsonized a bacteria to get that bacteria to stick. Remember also, all of these receptors are just proteins, right? So they're coded for by genes inside of, for example, the macrophage, right? And so these proteins that are made are very specific shapes so that when they're on the surface of the cell, they will bind a particular group of related targets or families, whether they be carbohydrates, other proteins, or things like lipids. All right, let's start off by looking at something like the Mannose receptor. Oops, sorry. All right. So Mannose receptors belong to a general family of carbohydrate binding receptors, right? So these are proteins that are on the surface of the cell whose job it is to recognize other shapes that happen to be carbohydrates. So we call any protein that kind of can bind a carbohydrate as part of its function a lectin, right? So lectin is kind of a general term. So macrophages use lectins to bind carbohydrates that are on the surface of bacteria. Now, as you might imagine, evolutionarily, it makes sense that we would have evolved to have particular receptors that recognize signals on bacteria, carbohydrates on bacteria, that you might normally not find on the surfaces of the cells of your own body, right? Because if you can preferentially recognize and bind something that's more unique to the surface of a pathogen, then that receptor is more useful. So in this case, the sugar mannose, you see the structure here, it's very similar to glucose. I think really the only difference is, well, I shouldn't say the only difference, but glucose has, what is it? I think the structure of this is very similar, right? It's a 6-carbon ring sugar, right? And mannose is slightly differently shaped than glucose, and that slight difference makes all the difference in terms of something that's going to recognize it. So the most primitive forms of mannose receptors are things like dectin. Right? So again, the book, the notorious book for oversimplifying or dumbing down molecular structures, right? Here's a good example of that. So dectin is represented here as this kind of blue Pac-Man, and then there's a stalk that sticks through or anchors the dectin protein to the surface of the macrophage, right? So this is how the dectin would be planted on the surface of the macrophage. But keep in mind, right, it's actually a very complicated 3-dimensional shape, right? So all receptors, all cell surface receptors have that feature of being anchored to the cell membrane. Right? There will be one part of it that sticks out, and that's the part that's going to do the receiving part, right? That makes it a receptor. And typically, there's going to be a cytoplasmic part of that protein that does the signaling. Right? So if something binds to the outside, then the part that's on the inside of the cell, right, passes on a signal. All right. How do proteins pass on signals? They pass on signals by the function of them changing shape. Right? So if something binds to the blue Pac-Man part here, right, it changes the shape of the protein at that part, then there's sort of like a ripple effect where the rest of the protein kind of has to change its shape to get comfortable again. And that will result in the cytoplasmic part of the protein also changing shape. And now that new shape might allow other proteins that are inside the cell to suddenly interact with it, right? Whereas prior to the thing binding to the outside, right, the shape of the receptor was not compatible for things inside the cytoplasm to interact. Right? Okay, so dectin binds to mannose, right, on the surfaces of pathogens. Here's another receptor that is just called Mannose receptor, right? So that's kind of an unfortunate name here because technically Dectin also binds Mannose, right? But this one got the privilege of being called Mannose receptor. So you see, this one is quite a bit more elaborate, right, than the simple Dectin receptor. This one has several Pac-Men here, these green Pac-Men, which are all Mannose binding domains, right? So already you can tell, yeah, this thing can bind much more Mannose that might be on the surface of a bacteria, right? And here's an interesting feature. The very end of this protein has this, what we call ricin type binding domain. Right? So ricin, what's ricin? Right? You might have heard of it before, but you thought, yeah, I think I've heard of it, but it's like a bad thing, right? Yeah, ricin is actually a very toxic protein. In fact, it's one of the most toxic small molecular proteins that we know of. Right? So ricin, maybe if you've heard of it, it's in the context of people doing really bad things with ricin, right? Like using it as a biological warfare agent. And there have been, unfortunately, incidents of terrorist attacks, like domestic terrorist attacks, where people have released ricin into the air of places like, for example, subway trains or subway stations. That's what happened in Japan many years ago, right? So how does ricin work? Ricin is a protein. It's made up of two sort of parts. You can see here it has this blue polypeptide domain and this, yellow or orange colored one in this figure here. Right. One of these parts is in charge of binding to galactosamines that are on the surface of human cells. So galactosamine is simply what? Some sort of a protein, that's the amine part, that also has some galactose sugars on it. Right. So that's why I should remind you, right, that our cells on the surface do have many sugars as well. Right. So things like these galactosamines, right, are on the surface of our cells normally. Right. So these little sugar groups, these galactoses, and the ricin protein, part of it, right, is the right shape to be able to bind to that. So if a ricin molecule arrives at the surface of one of your cells, right, like it got breathed in or something like that, this part of it will attach to the surface of the cell, and the ricin protein will then move into the cell. Once the ricin protein moves into the cell, it falls apart, and the other part of it, right, is the really dangerous, bad, toxic part, because the other part of the ricin is the right shape to actually bind to the ribosomes inside of your cell. Remember, the ribosomes are these large multi-protein factories that take care of translating, right, messenger RNA that has come out of your nucleus, translating that mRNA into a chain of amino acids, which create your proteins, right? So this ricin, the ricin part, binds to the ribosome and it stops the ribosome from working. And if that ribosome stops working, right, then it cannot produce any of the proteins that your cell normally needs to be producing all the time. How toxic is ricin? It's super toxic, right? Because they've done research to show that one ricin protein entering a single cell can bind to and disable up to 1,500 ribosomes per minute, right? And of course, if you're exposed to ricin, you're not going to be exposed to just one molecule of it, right? So therefore, it is super, super deadly because your cell, once it has its ribosome shut down, the cell essentially instantly dies. Okay, so what does that have to do with this interesting receptor on the left? The Mannose receptor has part of its domain that is very similarly shaped to the part of the ricin protein that actually takes care of binding galactosamines on the surface of your cells, right? Fortunately, right, it doesn't have the other part of the ricin molecule, right? So why would you do that? Why would you care that this top part of this Mannose receptor can bind to the surface of your own, for example, macrophage, right, that has this on it? It's because if you imagine the size scale of this single Mannose receptor that's shown here, kind of looks like a rose, right, with leaves and a flower, right, compared to the size scale of something like an actual bacteria that's coming in, right, and pressing up against the macrophage. I mean, the bacteria is huge, right? If this was actually the size of an individual mannose binding domain, right? Mannose receptor shown here, then a bacteria would be the size of like a blimp, right? So as the bacteria presses up against the macrophage, what will happen is that the mannose receptor will bend over as it's being squashed, allowing these green Pac-Men to have multiple chances to now detect mannose on the surface of the bacteria, right? Now, if the bacteria really is the size of a blimp compared to this individual mannose receptor, right, the mannose receptor might just tear right off the surface of the macrophage. But as the receptor is bent over, what happens is that the ricin type binding domain ends up getting pressed back to the surface of the macrophage, right, where it can now bind to the galactosamines that are on the surface of the macrophage, thereby essentially bracing or anchoring the receptor to make it a lot stronger and sturdier. All right, so if you have thousands upon thousands of these mannose receptors all being bent over with multiple mannose binding sites and being anchored to the macrophage, you can now imagine that now the macrophage has an easier time clinging to the bacteria and eating it. All right. Okay, here's another group of very important receptors that can detect common, generic, everyday pathogen signals from something like a bacteria. Remember, we mentioned that many bacteria, their outer coatings are a thick layer of polysaccharides, right? So they have these lipopolysaccharide molecules, so a lipid part that keeps them as part of the cell wall of the bacteria, and then these long chains of simple sugars, right? I said it's just like shag carpeting that ends up hiding little things underneath, right? It's like when I walk into my kids' room, they have shag carpeting, and I'm always stepping on Legos that are hidden in the shag carpeting, right? Until I step on them, I can't see them. So in the same way, certain proteins that are on the surface of a bacteria that could serve as good targets for your immune system to see might be hidden in this thick shag carpeting of lipopolysaccharides. But if you think about it that way, that means lipopolysaccharides, or LPS, are a pretty generic common thing that our immune systems would have evolved to be able to detect as being sort of a sign that, oh, maybe this is a pathogen, right? So let's look at this. Here we have a bacteria that's covered in lipopolysaccharides. It's like a shaggy dog that's just shedding, right? So lipopolysaccharides fall off. In our tissues and our blood, we'd have these LPS binding proteins, this sort of generic purple shaped thing here, right? And then this purple thing will bind to these lipopolysaccharides, and it will bring it to the surface of a macrophage, where the lipopolysaccharide will be passed on to this other green receptor known as CD14. CD14 is the right shape then to come over and find this big yellow receptor, which is known as a toll-like receptor, right? In this case, TLR4, or toll-like receptor 4. Right? What will then happen is there's another protein known as MD2, this like blue one, that will basically take the lipopolysaccharide, right, and now make that part of the toll-like receptor structure. That will cause then the toll-like receptor to change its shape. And as it changes its shape out here, there's a ripple effect, right, that causes the inside part of the toll-like receptor that's sticking into the cytoplasm of the cell to change its shape. And then that will then start attracting other proteins and kick off a series of communication. So here's the actual space-filling model of the toll-like receptor. You can see it actually functions as a dimer, right? Two of these sort of crescent-shaped TLR molecules work together. And then here in light blue is the MD2 that's hanging on to the lipopolysaccharide. So you can tell here, right, this is a chain of simple sugar rings. That's the LPS. And together, this is changing the shape of the toll-like receptor so that this part that's on the bottom here, right, changes shape and now might create something like this particular pocket that proteins inside of the cell can now interact with. Right? So why are these called toll-like receptors? Right? Because it sort of implies that this receptor looks like something that's already been called a toll receptor, right? So it's this toll-like. That's because these receptors were originally identified in Drosophila, or fruit flies. I think you all know that fruit flies are a very favorite animal model or insect model for geneticists, right? Because they're easy to manipulate and you can sort of identify mutations and things. So the story is that these were actually identified by a German cell biologist, Christiane Boldhart, and she was studying different types of receptor proteins to try to understand how different receptor proteins might cause changes in cells in response to different types of things, including things like pathogens, right? And so she discovered that these receptors, when they bound to the presence of lipopolysaccharide, trigger immune responses in the cells. And the story goes that her reaction to this upon seeing this in the lab, right, was das ist ja toll, right, which means in German, I know I totally butchered that. So for any of you who speak German, sorry, right? But that's basically, that's amazing, right? So that's why originally in the flies, these were called toll receptors, right, or amazing receptors. Later, when researchers realized that even in our cells, on the surfaces of our cells, we have receptors that are essentially, right, evolutionarily homologous to these toll receptors in flies. That's why they named our receptors toll-like receptors, or TLRs. All right, so here's an illustration of how these signaling events take place, right? So again, lipopolysaccharides that are being shed by bacteria, right, are basically transported by proteins like LBP to the surface of the macrophage, where CD14 passes it on to the MD2 protein inside of the TLR4 structure, right? So then the TLR4 protein changes its shape, right? So inside this, the cytoplasm, this part changes shape, and suddenly it's the right shape to interact with other proteins like this MyD88, right? That previously would just maybe float by or bounce off without actually attaching. So then the MyD88 kicks off a whole chain of other proteins talking to each other. Now, don't worry, you do not have to memorize this pathway, but I do want to show it to you because it's important that you understand, right, that when things bind to receptor proteins on the outside of a cell, right, they cause the receptor proteins to change shape, and that kicks off a whole bunch of other proteins now being able to interact with each other. And that represents the signal that ends up getting into the nucleus of the cell. And in this case, one of the important proteins that I do need you to remember is NF-kappa B, right? Because NF-kappa B is a very important transcription factor protein that's used for many, many cell functions. In this case, right, NF-kappa B gets activated and it binds to the regulatory sites of different genes, right? Genes that up until now maybe were shut down, were not being expressed, but now they get turned on, right? So the genes get expressed, you get mRNA from them, that mRNA then gets translated by ribosomes, the green things here, to make the finished protein product. And in this case, it's these little peach-colored triangles that get then moved out of the cell and released. And these are the cytokines, right? Okay. So we said that our innate response then has these two sides of defense, right? The immediate response and then the induced response. So let's look at some of the features of this induced response. We already mentioned that things like inflammation is a big part of it, right? But let's look at some examples of cells that are involved, right? Things like neutrophils, for example. Right? So what are neutrophils? So neutrophils are another example of leukocytes, or white blood cells, and they are the most abundant white blood cells in your system. Right? You have billions upon billions of neutrophils coursing through your blood right now. Right? In fact, it's estimated that an adult like you or me, right now you have something like 10 to the 11th neutrophils that are being released by your bone marrow into your blood every single day. Right? And a microliter, teeny tiny microliter of your blood should have on the order of 5,000 to 8,000 neutrophils in it. Right? If you have, let's say, 1,500 or below, right, then that's a reason for alarm. So oftentimes if you do lab tests, like if you do a physical exam and they do labs, right, on the printout, they'll indicate things like how many neutrophils you actually have, right? And if you're below 1.5, meaning below 1500 per microliter, then that might be indication that something's wrong, right? Because you really should have a lot. So in our bone marrow, we have even more neutrophils, right? So most of the neutrophils are actually hanging out in the bone marrow. When they get released into the blood, they only last for something like a few hours, like 5 or 6 hours, before they end up just being recycled. They're actually destroyed by places like your spleen, right? But if neutrophils, right, are released from your blood, released from your bone marrow in response to rising levels of cytokines, right? Because remember, the cytokines are being released by macrophages as they continue to fight the bacteria at the site of infection. Right? And as those cytokine levels rise, right, you begin to get more and more neutrophils being released by your blood, by the bone marrow into your blood. Right? And those neutrophils in your blood that are passing by, they then stop at the site of infection, squeeze their way out, and join the macrophages in this kind of battle here. Right? So you can see the macrophages were already there at the start. They were trying to eat bacteria, but maybe they weren't doing a great job, right? Like in this cartoon, for example, right? These 3 macrophages, you only see one of them represented as actually eating a bacteria, right? So that's why this infection has now gone from a few hours to a few days. Now you have this induced response. Lots of neutrophils are coming, and these neutrophils are far more effective at phagocytosis. However, each individual neutrophil can only eat something like 1 or 2 bacteria, and then that's it. They've done their job, right? They die, and you can see the macrophages then have to clean up the mess, right? So these sort of cells with these dark spots in them are actually dead neutrophils that have killed the bacteria, right? So the remnants of the bacteria are also in the neutrophil. The macrophages have to clean up this mess, right, of all these neutrophils. So how do neutrophils know where to exit the blood? If they're circulating around in the blood, right, consider this. At the inside of your blood vessels, if you think about it like a whole network of highways and roads, the inner wall of your blood vessel kind of looks pretty generic no matter where you are, right? So if you imagine a neutrophil as being a car, it's like driving through a whole series of tunnels, right, where you can't tell where you are, right? So this photo, when I was a little kid growing up in Boston, the airport in Boston is Logan Airport. It's actually built out in the bay. And so to get from sort of the mainland part of downtown Boston to the airport, you actually go through a tunnel that goes underneath the bay or the harbor, right? And as a kid, I was amazed by this because I loved it when we'd go into this tunnel and then you're like going through this tunnel for a long time, right? So if you think, for example, the H3 tunnel is a long tunnel, right? These tunnels in Boston are actually quite longer, and they're more extensive because they actually branch, right? They have things like exit points, right? But how would you know that you need to exit to get up to back to the ground level and get to the airport, right? Well, the tunnel, of course, has signs in it to let you know, right? So this is where you're supposed to exit over here to get to Logan Airport. So that's the same thing that takes place inside of our blood vessels. The endothelial cells, in other words, the cells that make up the wall of the blood vessels, will display certain signs that allow neutrophils or other cells that are passing by to know when to stop and exit. Okay, so these types of signals, again, involve interactions between receptors. So, for example, neutrophils on their surface have a particular receptor known as a vascular addressin, or CD34. This is the shape of it here. It has these special sugar groups on it, which are called Sialo-Lewis X sugar groups. You don't have to really know what that technically means. It's a bit O-chemy for this class. But keep in mind that that's the feature that they have, these special sugar groups that serve as sort of a way to attach to something else. The endothelial cells, right, the lining of your blood vessel, will express particular signs, for example, this selectin, right, that will then bind to the addressin of the neutrophil. And that will quite literally cause the neutrophil to start sticking to the wall of the capillary, right, or the blood vessel. Here's another example. You have things like the neutrophil that express these integrins on their surface, like LFA1, right? And these bind to this particular mark, this ICAM, right, or immunoglobulin-like molecule that's expressed on the surface of the endothelial cell, right, allowing there to be a tight bond with the neutrophil. Keep in mind that these selectins or ICAMs normally wouldn't be displayed on a neutrophil, right, if there's no infection going on. But if there's an infection going on, then these endothelial cells start displaying these signs so that neutrophils passing by, right, can then see them and know when to exit the tunnel, essentially, right? So here we have an example. We're looking at the sort of cross-section of a capillary. Again, these purple cells are your endothelial cells. The wall of your blood vessel capillary is very thin, just one cell layer thick. So below it in light blue is the underlying tissue, right? And let's say that down here in the light blue tissue is where the infection is. So maybe this is the site where you got a cut or something. So you don't see it here, but under here you'd have macrophages, right, and bacteria sort of battling it out. So because the macrophages have released cytokine messages, these endothelial cells, the purple ones, receive those cytokine messages, right? And that stimulates them to turn on the expression of things like selectin. So as these neutrophils tumble by, right, they begin to stick to these selectins and they slow down. Right? All right. So if this involves things like cytokines being released from the macrophage that's causing these chain of events to get neutrophils to come, what are some of these cytokines, right? I mean, yeah, they're generic, colored triangles as far as the textbook's concerned, but let's figure out what these are. So here are some examples of cytokines that induce inflammation, right? And cytokines that induce inflammation are what we call interleukins, right? Or IL proteins, right? For interleukin. The name interleukin, you can just take it apart. It means what? Two different leukocytes or white blood cells communicating with each other. This is a message between two leukocytes, or interleukin, right? So here are some examples of interleukins that cause inflammation. We will look at these in turn, all right? But let's start off by just looking at this example, CXCL8, because when the macrophages release CXCL8, this is what recruits neutrophils from the blood and guides them to leave the blood and go into the infected tissues. Okay. So CXCL8 is shown here. It is a very short protein. So the whole protein sequence is shown here. And they're called CXCL8 because all of them have in the middle, right, these two cysteine residues, with something in the middle. In this case, you have a valine, right? But it could be some other amino acid. That's why we usually use X, right? Just to show that has this feature, right? So these cysteines form disulfide bonds with other cysteines in the protein, thereby wrapping up this little protein into a particular three-dimensional shape. And that's the shape that you see here, CXCL8, right? We also call CXCL8, because it's an interleukin, we also call it IL-8, right? And so these protein signals then are received by neutrophils, and they get them to leave the blood. So let's look. The neutrophil has come to a stop because it's stuck to these selectin molecules. Right? These endothelial cells are now also expressing ICAMs. So these ICAMs also now are attached to the LFA integrins that are on the surface of a neutrophil. And this really now anchors the neutrophil, right? So it's tumbling, and now it comes to a stop. And now the neutrophil begins to smell the scent of CXCL8. Right. So down below in this tissue in the blue here, you don't see it happening, but there are macrophages that are releasing these green dots that are the CXCL8, right? And they drift away from the site of infection, reaching the capillary here, right? And then the neutrophils have receptors that bind to the CXCL8. And what that does is it causes the neutrophil to begin to change its shape, and it begins to change its shape towards the direction of where these CXCL8 signals are coming from. That's how the neutrophil then squeezes out of the blood and begins to make its way into the tissue to get to the source of these cytokines, which of course would be the macrophages that are battling the bacteria. Okay, we're almost out of time, but I just have a couple more slides. So let's just go through this pretty quickly. The neutrophils, as I said, are very strong phagocytic cells, right? They're much more phagocytically active than macrophages. Inside of them, they contain lots of vesicles that have all kinds of enzymes in them that can digest proteins, right? Importantly, these enzymes are normally held in an inactive state as proenzymes, because maybe the shape of the enzymes, right, are such that they're not active, these proteins. But when the neutrophil begins to take in bacteria, like you see here, right, the neutrophil exhibits a very bizarre phenomenon known as a respiratory burst. What it does is it increases the cellular metabolism by 100-fold, right? So it's normal sort of cell metabolism functions, right, that are producing some sort of like acidic waste products and stuff like that. Normally the cell can manage that, but now it suddenly ramps up this metabolism by 100-fold over the course of a minute or so. So that means suddenly the pH environment inside of the neutrophil goes all out of whack, right? I think it swings one way and then the other. I think it becomes very acidic first, and then it becomes very basic. And that acidity, that change in pH, right, suddenly causes these enzymes to activate. And then these enzymes go to work and they digest the bacteria, right? But unfortunately, these enzymes are so powerful that it actually ends up digesting the neutrophil itself, right? Because the neutrophil is not designed to be able to handle all of these crazy enzymes that it itself made. So both the pathogen and the neutrophil die as the result. And as we saw on a previous cartoon, then the macrophages are left having to clean it up, right? So as these neutrophils are dying, though, they have one more trick up their sleeve, right? Because I said each individual neutrophil can only maybe phagocytose 1 or 2 bacteria, right? But when this neutrophil begins to die, right, instead of just shriveling away, right, it actually explodes, right, and spills all of its contents out into the surrounding tissue. That includes lots of these enzymes that it had activated inside of it, and importantly, all of the genomic material inside the nucleus of the neutrophil spews out in a big sticky net, right? Because the chromatin is very sticky, and it'll carry lots of these enzymes and drape itself over many bacteria, right? Look at this image on the left, right? So these are all strands of chromatin from an exploded neutrophil, and they're covered with these terrible enzymes, right, that then get draped over the bacteria. And so the dying neutrophil, as its final act, doesn't just take out 1 or 2 bacteria that it ate, it can take out several dozen, right? So we call this a neutrophil extracellular trap, right? I kind of think of it like a kamikaze pilot, right? Like in World War II, when the fighter jet is going down in flames, right? The kamikaze pilot tries to target and ram the jet into like an aircraft carrier or something like that. And that's what our neutrophils do as well. So what does that result in? It results in a big mess, right? A big mess of all kinds of dead neutrophils that the macrophages have to clean up, and this sticky, stringy chromatin. And that, you already know what that is, right? That is pus. So pus, right, is made of billions and billions of dead neutrophils, and it's sticky because of all of this chromatin, right? And of course, all the dead bacteria too, that the macrophages then have to sort of clean up. All right, so that's it for today. This last slide just shows you, right, an image of infected tissue, and all of these dark purple cells are neutrophils going to work. Okay, this little cross section here is a cross section of a capillary. That's why you see some red blood cells that happen to be here. But I want you to notice is in between these neutrophils, see all the wispy, stringy stuff, right? Those are the remnants of exploded neutrophils up here as well, right? That are doing a great job of taking out the bacteria in large numbers, but resulting in some damage to your own tissues and the presence of nasty pus, right? Okay, let's go ahead and end there today. Sorry, I went over a little bit, but I'm happy to go to the chat if you want to stick around, and I'll see if I can answer some of your questions. All right. Okay, here's a question. So Conrad's asking, how do the membrane attack complexes and complement C3 interact? Can they plug the pores by chance? Okay, so the formation of membrane attack complexes starts by the activation of C3, right? And so when C3 is converted into C3b, remember we said at that moment, the C3b can either attach to the surface of the pathogen and contribute to opsonization, or the C3b can assist in the activation of complement C5 that leads to MAC. So I'm not sure what you mean by interact. They happen at the same time, right? Because we're not talking about one individual C3 molecule, right, choosing to go one way or the other way, right? If you have millions upon millions of C3s all choosing to go one way or the other way, you effectively have both events happening at the same time, the coating of the surface and the formation of these membrane attack complexes, right? Okay. So a follow-up question, the neutrophils are hoping to kill more bacteria than our own cells. Yes, absolutely, right? The neutrophils don't attack our own cells, but unfortunately, right, a big induced neutrophil response can lead to damage of our surrounding tissues. And all of us have experienced that, right? Because that is when you have a wound that has a bit of pus in it or something like that, right? There is tissue damage. But of course, the point is that that amount of tissue damaged, right, is worth it if it means you've put a stop to the infection, right? Because if you don't put a stop to the infection and the infection grows, you might die from it, right? So evolutionarily, we accept the fact that neutrophil activity, right, can damage some of our tissues, and we just live with the fact that we might end up healing up with a scar or something as a result of the damaged tissue. But at least we go on living, right? That's the point. We survive the infection. Okay. All right. So it looks like that's it for the questions. All right. So have a great weekend, and I'll see you on Monday. All right. All right. Bye-bye.